Hello and welcome to this webinar titled Developing Practical Single-Use Processes for New Vaccine Formulations, hosted by Biopharma Asia magazine and presented by Kirsten Strolendorf and Dr. Helene Pora. My name is Sri Patel and I will be the moderator. Before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now please allow me to introduce you to our presenters. For the last 12 years, Kirsten Strolendorf has been working as a senior scientist in research and development at Sanofi Pasteur in Toronto. She holds a professional engineering license and an honours degree in biological engineering from the University of Guelph and a master's degree in engineering from the University of Toronto. Kirsten takes a seat on the board of directors for the Bioprocess Systems Alliance. A driver in the bioprocessing community, she has published several journal articles. Kirsten manages a bioprocess design and scale-up laboratory for vaccine formulations. Her focus lies in automation and isolation systems for novel biotechnology products decades away from being marketed. Dr. Helene Pora is Vice President of Single-Use Technology within Por Life Sciences, where she has the global responsibility for the marketing and development of Por's single-use technologies. Helene has been instrumental in the development of Por's single-use technologies for the last 20 years. She is as well heavily involved in quality and regulatory aspects of single-use technology. Helene has over 30 years of experience working for the biopharmaceutical industry, the last 26 years within Por Corporation. She speaks regularly at conferences about single-use technology with a strong focus on validation and overall process integration aspects. She is as well involved in different working groups in the industry involved in standards development and is on the board of the BPSA and the SUTAP. I would now like to invite our first presenter, Kirsten Strolendorf, to join our webinar. Welcome, Kirsten.
Hello, and thank you very much, Sri. I would like to say thank you for inviting me to speak. I'm delighted and honored to be speaking to you today, and it looks like we have quite a global presence with a lot of people listening. So I look forward to presenting to you all and to any questions you may have afterwards. So I'm going to be talking about developing practical single-use processes for new vaccine formulations. To begin, I will go over an introduction, our objective and scope, and some formulation processing case studies involving particulates, interactions with gamma-irradiated assemblies, filtration, and integrity testing, all as they relate to single use. And then following that, some conclusions. I'm located in Toronto in our Bioprocess Research and Development facility. In fact, this picture on the left is our original building at the Sanofi Pasteur campus here in Toronto, where in 1913, Jerry Fitzgerald, a professor of hygiene from the University of Toronto and Pasteur Institute, used his wife's inheritance to build a backyard laboratory and stable. You can see the two buildings here on the left. Here he bought horses and used them to prepare diphtheria antitoxin. At the time, diphtheria was the #1 killer of children. And today, we still make this vaccine on site, but we no longer have the horses. So Sanofi Pasteur is one of the largest companies in the world devoted entirely to human vaccines. We're a division of Sanofi. We have a revenue of almost $5 billion as per 2015. Over 1 billion doses per year is what we produce. And some of our products, in addition to the diphtheria, include pertussis, typhoid fever, polio, influenza, and many others. Our headquarters are in Lyon, France, and we are a promoter of single use in development and industrial operation capacities across our sites. As single-use technologies have been a hot topic for several years now, and their advantages well known, I won't go into detail as to those advantages, but certainly the main ones would be greater sterility assurance, easy product changeover in a multi-product facility. Processing can be done in lower classified areas. I used to joke when we first introduced single-use technologies that we could run our vaccine formulations in a parking lot using the closed systems. Fast installation time, so it reduces our preparation work, which includes washing and autoclaving, assembling, and focuses our staff expertise on the actual formulation. Reduces potential for finding glass in final product. It reduces the cost of capital, yet it does increase the cost of consumables. And it reduces validation loads. So in vaccine manufacturing, single-use technologies are used throughout the processing stages from cell culture all the way to filling. And I won't go into detail here, but we've got our cell culture. After that, we have our cell harvest and capture clarification stages, purification and concentration, formulation and filling. And throughout that, our media and buffer prep. In all stages, we use single-use technologies. For our discussions, I will be focusing in the areas of formulation and filling. In terms of our manufacturing and design scope, this is for GMP manufacturing for human injection, but it's small-scale vaccine parenterals because we work from concept to phase 1-2 and sometimes to phase 3 manufacturing. But up to phase 1-2, there's no process validation. We deal with small batch sizes of 2 to 5 liters, or about 500 to 5,000 units, such as vials or syringes. In terms of the processing stages, it includes filtering, ingredient addition, adjuvantation, mixing, and resuspension, sampling, aliquoting, and transferring. All our systems are closed, single-use, gamma-radiated. There are many considerations during the selection stages, as many of you are probably familiar with, such as familiarity with the parts and materials, custom assembled, the fact that they're custom assembled by the manufacturer. What is the validation data available in the supporting documentation, such as extractables, gamma compatibility as per testing, gamma compatibility and testing as per the BPSA testing matrices? Are the materials transparent or semi-transparent? It is advantageous when the tubing and connectors and containers are transparent. Benefits of and material compatibility of the bags, filters and tubing materials, the pore sizes for filtration and throughput, scalability, the operation and storage temperatures, sterilization. We have a preference for animal-free components, latex-free, bisphenol A-free. We have to consider the sampling and interfacing. So how is it going to be connected to the next stage of processing or the former stage of processing, and the pricing, of course. So formulation case studies. Here I just wanted to show an example of one of our formulation processes. This is a multi-component vaccine. Direct adjuvantation formulation is what we call it. So we've got our ingredients all on the left, including the adjuvant, but all the ingredients on the left of the filters can be sterile filtered into the final bulk container, which you can see on the right in the rainbow colors. And we also add the adjuvant, which is pre-mixed, and added into the final bulk. At that point it's mixed and then sent off for sampling, for testing, and filling. All of this is done using a closed gamma-radiated assembly. So the first part of my case study is going to be with respect to particulates. And I feel like poor Sam here back in 2006 when I first launched single-use systems in our manufacturing facility. And there weren't really standards or guidelines readily available to follow. Particulates can become complicated in final drug product as they can either be intrinsic or extrinsic. Particulates can originate in the vaccine product, such as aggregation from the product in contact with the single-use materials, with respect to compatibility and or from the single-use components and assemblies, vaccine processing, environment, and packaging and storage conditions. There's a lot that could potentially generate particulates. These suppliers can send some information on the particulate levels for their particular products or assemblies, but it won't necessarily represent the full assembly that is going to be used with the product and the equipment that is used in the end user's facility. So here we show one of the particulate studies we did in-house, and you can see up above that we look primarily to see what the single-use provider can provide with respect to the particulates testing. And it doesn't just necessarily mean USP 788. It can be some of the other methods, the other pharmacopoeial methods as well, such as the European pharmacopoeia. But we also test with these limitations under our own worst-case conditions. And here we see a diagram of the particulate sampling throughout our stages of adsorption, final bulk, and filling manufacturing. So that includes all the stages that I mentioned before: transferring, mixing, and filling. So this manifold was set up to capture all of these stages. And of course, it's a single-use closed system. The components themselves, many of them were from Life Technologies, which is a corporation as part of Cyclone Thermo Fisher. This contains the ingredient containers, filters, and sterile connectors, welds, and processing lines, all worst case. And what I did was I put WFI, or water for injection, and drew it through the lines, collecting at various sample points. At those sample points, I sent them off to the lab where we could measure by light obscuration in accordance with USP 36, Chapter 788 for final bulk formulations with nominal volumes of greater than 100. The samples at volumes greater than 25 mils were aliquoted into 4 by 5 mil preparations for testing, where the first aliquot was discarded and the last 3 aliquots were measured to obtain a cumulative particle count per milliliter. And the limits for the USP that were used were at particle sizes greater than or equal to 10 microns, there are no more than 25 particles. At particle sizes greater than or equal to 25 microns, there were no more than 3 particles. I should also add that we also tracked the particle sizes that were smaller than 10 microns and larger than 25 microns as well, just to get an idea on where we were standing with respect to particle counts. You can see here, while we were trying to generate our worst case conditions, there's one case here where we had some leaking and some particle accumulations within the bag due to mixing under worst case conditions but poor conditions, and they had to be modified in our process so that we got reduced particle counts. So you can see in this graph we show this is before optimization, and this is the cumulative particle counts, and at the bottom you see all the samples themselves that we took at the various stages that were noted in the flow diagram 2 slides earlier. So you can see on the right the greater than or equal to 10 microns and greater than or equal to 25 microns and the limits of the particle counts there. So after the initial pre-optimized process, the counts were a little higher than expected in 3 main areas of the process. The first one was observed in many of the samples where syringes were used to withdraw the water samples by piercing them through a sample septum port on the bags. These were little sample bags, and we achieved irregular particle counts on these samples, but not on the samples that were not pulled from the syringes. The second problem was that we showed that the magnetic force generating the stirring mechanism on the single-use containers was too strong, generating the large particles. And you can see here from the last picture holes in the bags and causing leaking. The third was a result of the peristaltic pump that was used to pass the product, which was chewing the inner wall of the plastic TPE single-use tubing, leading to spallation and particulate generation. So after optimization here, we ran it again, and the WFI representing product that was passing through the assembly shown was exposed to all stages as it had been in the first trial. However, you can see there are fewer samples, and these were the worst case conditions and the ones areas of focus which we were looking at to reduce the particle counts, and they were still considered more worst case. The method of sampling, so the first problem was resolved by the method of sampling was modified so that the samples were withdrawn directly by opening the syringe port Luer lock connection on the sample bags. This was all done in the lab. The second one was a weaker yet still effective magnetic stirring mechanism was used in combination with capped mixing times, so fixed mixing times but not too long. A combination of optimized pump head, so the third problem was resolved with a combination of optimized pump head, tubing size, tension setting on the pump, and flow rate was applied. With these changes and the study repeated, all the particulate results passed the USP criteria. It should be noticed that we can still see a small increasing count for both the 10 and 25 micron sizes observed during the filling process, which are the ones on the right. You can see an increase there. And that can be explained by the fact that the peristaltic pump used to fill the product wears the platinum-cured silicone tubing ever so slightly over time. This was expected, and considering the process was run under worst-case conditions, and the accumulated count of particles at the end of the process was still well within the acceptance criteria. So moving on to my next case study, it's with respect to comparison of sterilization treatments of this particular Adjuvant A, which is an aluminum-based adjuvant. So currently we receive our Adjuvant A in a container that's already sterilized from the supplier. It comes in a bottle, has a cap, that gets opened and removed in the biohood. The problem with that is that it gets opened into a controlled yet open environment, and it's not ideal. We have been trying to close this process by sterilizing it in the container. So we are closing the process. In this study, we compare gamma radiation versus the stock untreated adjuvant. And when I say untreated, it's a little bit misleading because it has already been sterilized by the supplier. So I mean that it is untreated a second time. So we are comparing the stock versus autoclaved material and gamma radiated material. And we can see we have tested the gamma radiated material of the Adjuvant A in bags, which can be seen in the top right. So we use the high cone bags. And in the plastic mixing tanks on the left, these are the BioBlue containers that also come single use. And in a stainless steel tank, you can see here on the bottom right. It's a very small version of a tank. We ran characterization tests on the adjuvant, both qualitative and quantitative, to see if we could, in fact, sterilize the product without having a negative impact on it. But the first problem we saw was visually. You can see here on the left, after the product, the adjuvant was well mixed. You can see the stock solution, so that's our control, followed by the gamma radiated material that was, so the adjuvant itself was put in the bags and then sent off to be gamma radiated. Gamma radiated in plastic bottles and autoclaved in stainless steel. So you can see, might be a little difficult, but gamma radiated in bags, after it's been well mixed, looks a little foamy and emulsion-like, and unlike the other samples. And then, so we thought, well, why don't we settle it out and see if it has any impact? Let's look for that bilayer. So the stock on the right-hand side shows that nice bilayer, which is also reflected in after it's been gamma radiated in plastic bottles and after the adjuvant has been autoclaved in the stainless steel tank. But however, we see when the adjuvant is gamma radiated in bags, we see a strange looking uncharacteristic sedimentation and this clumpy bilayer formation. It raised some flags. By transmission electron microscopy, the worded description of fine needle-shaped crystals clustered into rosette-like aggregates was the same for all the samples. STK is stock. Bag is gamma irradiated in bags. Blue is gamma irradiated in the plastic BioBlue containers, and AUT is the autoclaved adjuvant. in the stainless steel. And you can see the pictures here. They all look roughly the same, except the gamma irradiated in bags looks a little bit more dense than the other samples. With respect to pH, you can see here that we have a decrease in pH of this sample that's been gamma irradiated in the bags, unlike the other samples that all hovered around 6.1 to 6.2. And you can see the stock reading at 6.23 was very similar to the stock CV, which was at 6.2 pH. And the specification is quite a wide range of 5.5 to 8.5. So all of the results met the spec. However, there was a notable decrease in pH when it was gamma irradiated in bags by almost 0.5. The next we tried was the adsorption. The adsorption of the adjuvant onto BSA. And I just wanted to point more to the y-axis on the left, which is absorption capacity. And you can see the absorption capacity, even though we've got quite a wide range of deviations here. We do see a decrease in the bag, gamma irradiated in bags, as well as autoclaved material. The autoclaved material after so this adjuvant, after it's been autoclaved again subsequently, it is expected to have a decrease of roughly around 10% in terms of absorption capacity. So that was expected, but the fact that the bag was lower was a little bit of a concern. It's actually significantly lower. I'm not showing all the results here, but what I did demonstrate was we ran a conical plot which gathers and ranks all the quantifiable test methods that we used to characterize the adjuvant. And we wanted to see which ones cluster most closely to the stock adjuvant, which can be seen kind of in the greenish yellow rings on the right. And what we see is that the gamma irradiated adjuvant A in bags does not plot in a cluster, as do all the other samples. So in conclusion for this part, ingredient compatibility, where the product is put into the containers and then sent off for irradiation, requires careful consideration. There is a change noted between adjuvant irradiated in bags versus all other samples, and this can be seen by visual observation and microscopy, statistically by quantifiable test methods, and by pH and absorption. The samples were taken for particle size, aluminum content, crystallinity, and PZC, but they show no immediate change across all samples. We actually are further evaluating what is changing between the gamma irradiated adjuvant in bags versus plastic bottles and different film types to see if this impacts our vaccine formulation. Overall, adjuvant gamma irradiated in plastic bottles and autoclaved in steel tanks provide a comparable option to untreated adjuvant. This was an interesting study on filtration. So in this particular case, we had a cationic peptide that was tech transferred to us within Sanofi Pasteur from another party. And this vaccine requires sterile filtration, as many of our vaccine components cannot be terminally sterilized. It required sterile filtration. So the old process involved transferring the ingredient, or forcing it essentially through a sterile filter using a high pressure with a maximum of 30 psi. A new process was needed, as the intended current GMP facility, in our facility, we had no qualified pressure tanks or systems to be able to apply this higher pressure. It wasn't suitable for single use, of course, as there's no single-use applications that can be rated with such a high pressure rating at the time of this filtration, and we didn't have any steel tanks available for use. In addition, there are specific safety precautions that must be taken into account using a process under pressure. And if it can be avoided, a single-use application is better. So what we did was a strategic method where we took QBD principles and applied them, such as testing potential critical quality attributes that were impacted by process parameters applied from the peptide dissolution to sterile filtration stages. These attributes included particle size and peptide concentrations primarily. In-process controls were tested, including pressure, turbidity, product sterility, and bioburden. However, for the purposes of the optimization stages, the three were tested after the process was defined and test methods were available and/or qualified. The first efforts were to sterile filter the solution by applying constant flow using a peristaltic pump through various filter membranes independently, and flow rates at small scale. These included membranes that were recommended to be used as they were neutral and slightly positive to prevent product buildup of the cationic solution on the surfaces of the membrane. Unfortunately, the dissolved material that was to be filtered had a tendency to form these interesting long filamentous networks that had large clumps aggregating on the filter membranes and clogging the filters very easily, such that the pressure readings across the filters using a baseline single-use pressure sensor was greater than 20 psi. The product recovery concentration by RPHPLC was reduced to approximately 57 to 75%, and volume recovery was less than 50% when constant flow filtration was applied on various membranes, though improved when the product concentration upstream of the filter was diluted. So diluting the material did help. Both pre-filter membranes built into the sterile filter capsules, as well as independent pre-filters of various porosities, were tried with little success. Such low recoveries and the fact that the pressure change was higher than what was feasible for the lab's typical single-use sterile filtration manifolds indicates this process was not feasible. So, in turn, analysis was performed on the material after gentle and vigorous dissolution methods were tried. In addition, we challenged using a glass beaker and single-use. So the single-use component was a plastic spinner flask container with a similar stir bar to flask dimension ratio as the glass beaker, so that we eliminated a variable. The material was dissolved into these containers, and they were stored for 1, 3, and 13 days. The mean particle sizes shown here by dynamic light scattering demonstrate that neither container material nor method of dissolution had a dramatic impact on the process. After 13 days, the particle size was almost doubled, as can be seen in the columns on the right. The storage time after dissolution resulted in the largest change. The recommendation was to try sterile filtration by constant flow a second time. But this time, solution was filtered within 1 day post-dissolution to avoid aggregation or filamentous networking. When passing the dissolved and more diluted solution through a dual sterile filtration immediately after dissolution, both product recovery and HPLC concentration and volume were 100% and were therefore improved with this process. In addition, a pressure change occurred of less than 2 psi using the Pendotech single-use pressure sensor and monitor. So this is a case where the product process was modified to accommodate single use, as filtration could not be implemented with applied pressure, and at the time the product results of interest were improved using this new method. So constant flow provides a better alternative to constant pressure in terms of safety, recovery, and throughput. Filtering as soon as the product is dissolved and diluting occurs is done in single-use constant flow filtration. My next case study is with respect to container integrity. So we do proof of concept runs, optimization, and confirmatory batches with single-use assemblies. And what we've noticed that if we can run integrity studies early on when we're doing these, it identifies gross leaks in the assemblies early on. There is a stage where we run a verification to see that the supplier and or we as end users have performed some of the necessary validation studies that are listed here. Some of them are destructive and some of them are non-destructive. I should note that the BPSA, the Bioprocess Systems Alliance quality testing matrices, is an excellent source for defining these tests. So our in-house container integrity study was done to prove that equipment can detect these leaks successfully with the end user's general assemblies. And when I say leaks, I mean gross leaks. So 3 integrity testers, A, B, and C, were tried, and they were the only ones that were on the market at the time. The parameters were set up for each unit and each assembly type. And that actually took some time to do, to set up all the pressures, time, and the general conditions to run the pressure changes. The leaks that were looked at include deliberate punctures made on the surfaces, at the bag seams, and at the junctions, at the connections or the endpoints. So you can see here we tried 3D, 3-dimensional, and 2D bags. We tried sterile connectors. We tried a single-use filling line with a needle and a complete assembled sampling manifold. All of these were gamma radiated prior to being tested. In summary, we can see the units in the columns A, B, and C. And what we see here are the controls. So we see where we've got ones where we don't expect there to be leaks and ones where we've made deliberate punctures. And you can see the size of the puncture holes. And in all cases, you can see where there were not supposed to be any leaks, then no leaks were detected. And where there were supposed to be leaks, there were leaks detected, except in the case below in the filling lines, where the leaks were not caught. So all 3 units generated a pass against the failed positive controls and the negative controls, as mentioned with the exception of the single-use filling lines. So we were thinking that when we punctured the holes in the filling lines, what was happening was as we passed air through it, it was actually causing the tubing to kind of swell and the hole to close in on itself. So it wasn't protecting the hole. The filling line has a very small inner diameter of 1 32 inches in inner diameter. I should mention that we had some difficulty creating the calibration holes and openings for the positive controls, which can be mitigated in future using some companies that actually sell these types of calibration holes. For us, we could not achieve a 30-micron hole at the time. But the testing time ranged from less than 10 minutes to 30 minutes, depending on the unit and the assembly. Also, we had to connect an adapter between the testing unit and the single-use assembly. In some cases, it was with a sterile filter, a vent filter, and in other cases, it was a single-use pre-sterile connection that was connected with sterile connectors. The filters and the vented lines had to be clamped off, which in some cases, you know, I guess that's one of the limitations of the machine, is that how those are clamped off using hemostats or C-clamps, you know, how far you tighten those, it can create a limitation because you're not testing anything downstream of that clamp, and you have to make sure that the clamp is done in such a way that it's not leaking itself. In some cases, the lines were sealed off, so it wasn't a problem. The test pressure was within 3 to 500 millibars, but of course this depends on the equipment, the size or volume of the single-use assembly, and the complexity of the assembly. I should note, Alain Porras, who's speaking next, has been leading the authoring of paper on integrity testing for single-use applications as part of the BPSA. In conclusion, our formulation and filling single-use processes are optimized and tested to ensure the outcomes are within specifications, including product performance and process outputs. In the case of the particulate studies, the entire single-use process was mapped and tested to find weak points where the particle counts were higher. These process elements were optimized, and then as well as the single-use assembly, and it was retested so that we followed within the USP criteria. With respect to compatibility after irradiation of the adjuvant A, the ingredient compatibility with the irradiated containers requires careful consideration, as changes may occur. With respect to the filtration case study, product-specific attributes and process outputs are measured to verify the recovery, and these can be monitored and controlled using single-use PAT. With respect to single-use integrity testing, end users can now test single-use assemblies non-destructively in-house for growth plates. Formulating and filling of vaccines in single-use have many aspects to test and design, as can be seen. Often, intended vaccine process components, equipment, and parameters may need to be changed to accommodate a single-use system. But my main point is it's the responsibility of the end user to test or have tested the complete single-use assembly in the intended facility, with users' representative worst-case parameters. And in some cases, you have to design the process around the single-use assembly, and in others, you have to design the single-use assembly around the process. And that ends my talk.
Thank you very much, Kirsten. So just as a reminder to our listeners, if you have any questions for Kirsten or for Dr. Pora, then please click on the questions tab at the bottom of the screen and send them through to us. And we will be asking these at the end of the presentation. I would now like to invite the speaker for the second half of this presentation, Dr. Helene Pora, to present an industry perspective titled End-to-End Single-Use Solutions in Formulation and Filling Process. Welcome, Dr. Pora.
Okay, thank you very much for the nice introduction, and I want to thank Kirsten very warmly because I think she's making my job easier now in order to present, because at least what everyone can understand is that there is true consideration from the industry about using single-use technologies in formulation and filling application. And I think that it's important to really understand that the end users are embracing single-use technology for that type of application, because what is really true is we understand that this is where the drug is at, I would say, its most critical stage. And as a supplier, we have to understand that basically the patient safety is and has to be our first concern. And basically, as we are so close to the patient, I mean, obviously, we have as a supplier to be some kind of fully aware about the regulatory requirements, to be fully aware as well of the safety of operators, and we need to ensure that we are not going to affect at all the performance of the drug product. So obviously, I mean, Kirsten's job is, I mean, heavily focused more at, I would say, product development. But our experience is really now that we are seeing more and more projects which are really dealing with the commercial manufacturing line, where our end users are using that type of technology. So really, I mean, what is clear is that now the technology, I would say, is mature enough or is ready to address the formulation and filling application requirements. So where we are with single-use technologies, I think that we've moved a long way forward. You know, I think that if we are looking a little bit some kind of a few years ago, we were really, I mean, end users would use some kind of individual components, you know, a few bags here and there, some sterile connectors and so on. Then, I mean, obviously, we moved to assembled system, and that's the type of things that Kirsten has been presenting to the whole assembly. But what we are seeing now is really, I mean, a requirement for, I would say, fully automated platform, and then, I mean, working, I would say, in a collaborative manner with the end users, with filling machine suppliers as well, in order to be capable of providing to end users end-to-end fully integrated solutions. So what does it mean? I think that it means, if we look at it from, I would say, a supplier perspective, that at the end of the day, what is happening now is there's a lot of shared responsibilities between the supplier and the end user. What it means from a relationship point of view means that we have to work, I would say, in a very strong collaborative effort and collaborative manner. And basically, the result for the end users is using more and more single-use technology. They are transferring more of what were their responsibility to us as a supplier, which means that we have to grasp and understand what it means and ensure, again, that there is no concern around the patient safety at the end. So it means that we have to put a lot of attention related to the quality of the product that we are providing. This can only be done by, I would say, implementing very strong quality by design approach, having very strong qualification package, and then we have really to work hand in hand and to support the end users through the design, through the development of the solution, and then through the installation and the validation. And obviously, the supply chain is key, and we have to ensure that everything that we do it within the right lead time. At the end user still has a lot of responsibility. We always have to remember that they are the people who are going to stand in front of the regulatory authorities, and we have to do our job perfectly in order to really enable them to do this in a very robust manner. So they will still need to perform the risk assessment, particularly at the solution selection. And what I mean by the solution selection means as well at the supplier selection. They will have to demonstrate that the drug product properties are not affected by the chosen single-use technologies. And for example, that's some of the work that Kristen did when she was looking at the impact of gamma radiation, whether she was using bars or bottles. Then, obviously, look at drug compliance. And then, the end user will have to demonstrate that the manufacturing process comply with the regulatory requirements. So how do we deal with it as a supplier? So basically, what we have done is we have developed a very strong package looking at quality by design. And basically, we've worked at developing a unique system which does allow us to store and to maintain all the information related to component validation, to the junction validation, to the application validation, and to the design through a configurator. But basically, all this is fully interlinked to basically ensure that there is kind of nothing left to some degree of approximation. Then, obviously, Kristen mentioned that she had to work in closed system. That means that we have to provide a sterile product. So this means there is a sterile claim according to ISO 1137 with a sterility assurance of 10 to the 6. Kristen again mentioned how Particulates is important. So that's why, we have developed a full approach as well to support that our system complies to USP 78.8 and as well USP 85. Basically, integrity again is very important. So basically we work with, I would say, a whole batch of tests, ensuring that all our bulk containers, critical components are tested at 100%. And then, obviously, and Kristen was mentioning, we have developed as well technology which do allow our end users to test the integrity of system prior use using flow measurement technology. And basically, as a company, we are capable of helping our end users as well through process-specific validation services. So this is really important to understand that full approach, because basically we need to have the whole story and we need to be capable of assisting our customers, I would say, from the beginning till the end and the full implementation. So I'm going to give you some ideas about how do we do it. So obviously the end user is looking at and applying risk assessment in order to choose the best solution. So obviously there are some, I would say, critical parameters, everything linked to the quality, to as well all the handling. I think that handling is something that we never speak enough about, but this is really important. And if you look at a number of papers or case studies which have been published in the industry, there is a clear indication that handling is important, can create issues, and therefore, I mean, it means that training would be very important as well. And then, I mean, obviously, when you look at the supplier assessment, I mean, I would say there is a lot of things such as, design, validation support, delivery time, security of supply. But I think that you need as well to understand when you look at end-to-end solutions, as well things like automation capacity, because as I mentioned, I mean, more and more end users are really looking for that type of technology. So what it means, we start to work from obviously the user request specification or URS, and I mean, we try to build our answer based on what is the application and the requirements from the end user. And then, when we work at automated solution, we really look at how can we help and build the adequate platform for automation, which means that in terms of skill sets in our team, basically, when we go to that type of complex project, we need to have a very strong process engineering team with all the right skill sets, but it means as well that the project management, which does ensure that the project education is going to be, I would say, extremely good. Does happen. I mean, usually, we use the terminology that in order to do that type of thing, you need to execute within specification, within budget, and on time. And these are really the driving words to the execution of the process of the project once we've been selected. So in the next few slides, I'm going just very briefly to show you what is the type of complexity that we can reach with single-use technology, which not always everybody is fully aware. And basically, this is the type of formulation and filtration modules that we have supplied to end user, which are fully automated. So here we look at the formulation module, and basically what it does is that the mixer is fully controlled by the HMI. So is the addition of the different ingredients. And then, once this formulated solution has been filtered, then they are transferred to the bulk containers. And as you can see, you have a high degree of automation, and ensuring particularly the accuracy of dispensing. And what we do, and you can see some example in the picture, is basically we design our frames in order that basically you guide the operator for reducing operator errors, and that's kind of extremely important for single-use technologies. So again, we apply the same principle. I mentioned that we were having a filtration module. So then, basically we have been capable of integrating the automation of the filter integrity testing, and basically that reduced all the handling. And then, we have the final solution then from the formulation to the final filtration. In order to do these type of things, obviously, bulk filling is very important. So then we are building a fully automated bulk filling solution, which basically, again, allow our end users to operate in a lower grade classification, whether it can be class B or class C. And we look at accuracy of filling, that's very important. And again, where we put a lot of focus as a company is on the ease of operation and how we can reduce the handling in order to minimize really operator errors. Then, same technology can be applied again to final filling operation. There, I would say the level of complexity in terms of the relationship does increase slightly, because now we have as well to integrate into final filling operation then the filling machine suppliers. So that now becomes what I would call a three-party partnership. And again, we have to speak about partnership and collaboration. I would say it's not so much anymore just, supplier and user relationship. And basically it means that as the supplier of the single-use filling line, we speak a lot to the filling machine assembly to understand how it is going to work. We do a lot of collaborative testing, ensuring that everything is going to work fine at the end of the process. So it means that we will work and in a collaborative way, and ensure that the filling performance are qualified. So we will assist everyone during the FAT and the SAT. We have developed as well good technologies allowing for the aseptic and easy transfer into isolator using bit bags. As a company, we have developed some form of unique filling needles, which are made of PEEK and which performance are equivalent to stainless steel needles and really allow for as well node ring formation. And then, the combination of all these technologies, really improve a lot the product safety and ensure that at the end, because you are using single-use technology, you will have no cross-contamination. And obviously, as I mentioned, through our quality by design approach, we have a very robust way to document the qualification, the sterility claim, and obviously the cleanliness of the whole filling line. So if you look at it from a project perspective, you see that it's quite complex. As I mentioned, it's not only the end user and the single-use supplier. When we go to final filling, then you need to have the filling equipment. And you see the different steps that we have to take as we move from working from the URS, then providing or designing the solution, then to the evaluation from the end user. And I think Kirsten described very well what is the type of qualification work that as an end user you would like to implement. Then, obviously we will do the FAT, and again, in full collaboration, then we will move to SAT. And as part of the SAT, it does involve as well training of the end user, and I think we never emphasize enough how training is important for successful implementation of single-use technology. And then obviously the validation, and I mentioned earlier in my presentation that as a company we are fully capable of assisting customers with validation. So in terms of qualification requirements, I think that a lot has been said, and what I want to say is re-emphasize again the fact that as you end users, you are moving more to single-use technology. I would say you are putting more in our hands. We are very grateful for this, but we fully understand what it does mean in terms of commitment for responsibilities and so on. And I would say it can be successfully done only with a very quality by design approach, which means as well as end user that how you do your supplier qualification is going to be critical for the success of implementation. So validation, of the solution of single use. Obviously one of the focus which is always very important, I think that Kirsten mentioned particulates, she mentioned integrity, but there is as well, as we all know, a very strong focus on extractable and leachables. And I guess that most of you are fully aware of the work which has been done by the people group, leading to some form of an industry approach and looking at how a method standardization could occur in our field. And I'm going to briefly discuss this in the next two slides. So basically, as a company, what we've been doing is that we have embraced the approach, described by BPOD. And what we have done is basically we have developed full capabilities in terms of testing our own products according to BPOD. And the data package are available for a number of components. And basically, the idea is that using the data sets which are available from the individual components, basically then you can use them for your extractable and leachable risk assessment. And basically the way that you do it is that you really look at what are your process parameters, what are the risks, what is the need for your characterization related to extractable leachables, and then based on the data that you have from the supplier, whether they are, I would say, what is available now or whether they are now BPOD aligned data, basically you would use the decision tree which is on the right-hand side to decide whether all the data that you have are good enough and satisfy your requirements, or whether the data, are insufficient, and whether then you need to determine a few and do additional process studies. So what do you really have to remember, around single-use implementation is, obviously single-use technology is a very good answer in a number of cases, and particularly what it can allow you to do is obviously reduce cross-contamination risk, but it can as well allow you to reduce the grading of the area where you want to work. I think that it's important now to remember that basically the technology nowadays allow you to implement fully automated single-use solution, and I hope I've been capable of demonstrating you that even in very critical applications such as formulation and final fill, basically we have the capability to implement that type of solution. And basically you need to choose a supplier which is capable of assisting you related to the qualification and validation. But I guess that everybody will agree now that single-use solution, is a technology which is ready now for efficient and safe formulation and filling processes. So with this, I would like to thank you for your attention and take any questions.
Thank you very much, Dr. Porat. So at this point, I'd like to begin the question and answer session. So the first question I want to ask is, are you using any particular standards which are specific to single-use technologies when implementing these technologies? And that's open
yeah, thanks very much, Sheree. I can answer that in terms of from my end user experience. There are limited standards per se, but lots of guidelines for single-use implementation. The best recommendation that I can make is, for the one-stop shopping concept is to look to the Bioprocessing Systems Alliance testing matrices, which has a massive spreadsheet, which is broken down by component. So single-use sensor, bags, connectors, chromatography, skids, etc., as well as the type of testing. So are we looking at physical, chemical tests, chemical, biological, functional, safety, and it's a huge matrix matrices which kind of points you to the right guidelines or standards that may be available for these individual tests. In addition to that, it really depends on what we're looking at. Are we talking about particulates? Are we talking about extractables, leachables? There's lots of things to, are we looking at tensile strength? So this matrices can help. But also there's the new draft by the USP, this 661.3 on plastic components and systems used in pharmaceutical manufacturing. So that does cover a lot of these aspects and can offer some further guidance. I don't know if Helen, you wanted to add to that?
No, I think that your answer was very comprehensive, Kristen.
Okay.
Great, thank you very much. The next question is, how do you ensure your systems are integral when you are using them?
So maybe
sure. Yes, go ahead.
Okay, Kristen. Yes, I think that it's a collaborative approach. So I think that it's a combination of what we do as a supplier from the beginning, going from component selection and testing, looking at how do we ensure that the assembly process is fully validated. This is what I meant when I was discussing junction testing. Then, about additional testing that we may perform as a part of the manufacturing process. And now you have more and more, I would say, sophisticated technology, which can go up to the use of helium integrity testing in case of a highly critical application. But it goes as well to the care that we take of the packaging, because we know that packaging is very important. And then how, when it does arrive at the end user, how the end user is being trained in terms of using the product. And then, if needed, depending, and this is all based on risk mitigation approach taken by the end user, now with the availability of integrity test device, which do allow for testing of the integrity prior use. So I would say it's a whole battery of things which leads to a very high quality product. I think that's how we have to look at it. And again, as Kristen mentioned, all these points will be fully discussed in the integrity test paper that we are currently working on with the BPSA.
Excellent.
Yeah, just to add to that, we, as the end users, we look at what the supplier already has done and what they can provide to us, whether it's on the C of A or as part of their development and validation. But we look at the entire assembly or the entire system that is considered closed, and we run an integrity assessment, which really evaluates the application. So looking at the stage of manufacturing, the storage conditions, the type of assembly, and as I said, what validation already exists by the manufacturers. And in some cases, we would run container closure integrity test, such as a dye penetration, pressure. We could do a helium, typically we would source that out. In the final stages of vaccine manufacturing, we also do aseptic broth simulation studies. We also do stability studies, which also test for, in a sense, integrity. We do CCIT at the end of our stability and look at various aspects that feed into the elements of integrity, such as sterility or any changes that may impact the integrity or the product in the container. And, in the case of individual components, we, again, look at the vendor validation data and we assess, do we need to do more validation? And that we look at on a case-by-case basis. But as Helen said, she's leading the authorship of a wonderful integrity testing paper that will be available shortly through the BPSA.
Excellent. The next question is directed at Kirsten, but if you need to add anything to it, Dr. Poro, feel free to. It says, hi Kirsten, in regards to the gamma irradiation study, what was the dosage applied to the solution in the bags and the solution in the bottle? Also, you presented a visible difference between the gamma irradiated bags and the bottle irradiated solution. Do you know what was the dosage distribution, considering that the solution had different thickness on the bag compared to the bottle?
That's an excellent question coming from somebody who definitely knows the challenges of gamma radiation validation. So this wasn't a validation study per se, and to answer the first part of the question, so the dosage could have been anything greater than 25 or less than 50 kilorays. The one that we actually used, if I recall correctly, was around 36.1 kilorays, and so that was the exposure that was applied in the chamber. In terms of the distribution, so again, this wasn't a validation study, and we didn't compare exactly like for like. So, you know, the head spaces in the plastic versus plastic hard-walled container versus the bags were different. The materials were different. I should say though that the volumes were the same. We used 200 milliliters, but the volume to the surface area was different as well. But again, it was a preliminary study to see if we could even do it, if it was even an option for us. I do believe that we can irradiate this adjuvant in containers, but we have to pick the right containers, and it has to be run in accordance with, you know, looking at such things as the distribution. We did not look at penetration in terms of ensuring that it was sterile. We were just seeing the characteristic impacts it had on the adjuvant itself. So very good question. Thank you for that.
I think there's another question following, I don't know if it's the same or following on, but somebody's asked, was the adjuvant sterilized by gamma into the bag, or was it just entered into a gamma sterile bag? That question makes sense at all?
I'm not seeing that one. Can you ask that again, sorry?
Was the adjuvant sterilized by gamma into the bag, or was it just entered into a gamma sterile bag?
That's actually a really good question, and how we broke the study down. What the goal was we wanted to, pour the adjuvant into the bag and then irradiate it, such that the adjuvant was also irradiated along with the bag or the plastic container. And we did have to work with our suppliers to do like a one-off order of bags on the BioBlue container that were not irradiated or gamma or beta irradiated, which is how they typically come. So we had to order them essentially so that they hadn't been treated. Then the solution got entered and then sent off to the irradiation facility.
Great. The next question, what is your strategy regarding extractables and leachables?
Yeah, so in short answer from an end user perspective, I could say, from a vaccine company, I could say we follow the BPOG protocol. However, in my area, I bring single-use assemblies into small-scale GMP manufacturing to support phase 1-2 clinical trials. And so it's brought in through a development phase and then, the small-scale GMP manufacturing before it goes into a licensed industrial capacity. So we stage the extractables, leachables that we need to do, such as first we look at what the manufacturer has in existence for extractables data and how it may apply to us. Are we using solutions and conditions that are bracketed by the extractables data that's already available? That's where following the BPOG protocol helps. But we assess if we need to do further leachable studies, which is more likely to be the case if the product, the product ingredients, or the conditions at which we use them are something kind of unique, or if the ingredients are a little harsh or stringent, and may cause some expected leaching. But at that point, we bring it into our phase 1-2 clinical manufacturing, and then once it moves on, then we look at more product-specific leachable studies as we bring it in for licensed manufacturing. I hope that helps.
Dr. Pora, is there anything that you'd like to add to that at all?
No, I'm fine.
Great. Let's go on to our next question. Are you aware of supplier data for sporulation? Is this available, or do users have to do their own experimentation?
Yeah. Okay. Sorry, go ahead.
Yes, I think I can take this one. So basically, you have to understand that the sporulation is a result of the interaction between the tubing and the given pump. So really, first of all, it means that you have to carefully select the right tubing with the right pump. I think that's kind of my number one comment. And then usually you will have really to run experiments to understand and ensure that basically you reduce as much as possible the phenomenon. And I think that's, the key point is really selecting the right tubing and then ensuring that the pump is well used, and so on. So that's very important.
Anything you'd like to add to that, Kirsten?
Yeah, I mean, in terms of the spallation, we do ask for it when we're ordering tubing, whether it's silicone or thermoplastic elastomer. We do ask for it from the supplier, but in my experience, you ask for it from one supplier and then you get it from another supplier, and you're trying to compare two different types of tubing that are maybe similar. But the conditions that they tested spallation were different. So one might have gone for a longer time or used a different pump head, a different size tubing, etc. So that's why we look at it a little bit more closely, and sometimes we do it ourselves if we want to compare across different types of tubing. So I hope that answers the question.
Great. The next question. Is it obligatory by GMP or FDA regulations to do integrity testing of 100% SUS bags before process?
The answer is no, and the answer is really clear. There is no regulation which pertains specifically to single use. So really, this is driven by, I would say, risk assessment, and I would say practices between the end user and the supplier. So really, I think it has to be very important. There is no mandatory request to do this for a number of reasons, because it would not be practical, the technology may not give you all the answers that you want, and so on. So that's why, I mentioned earlier in my discussion that how you deal with integrity is, I would say, a number of, processes, best practices, which lead you to ensure that at the end you have an integral product.
Great. Anything you'd like to add, Kirsten?
No, I think Helen did a great job answering that.
Great. The next question, I'm trying to see what's being asked here. The question says, SUS training support knowledge, support or knowledge management. I think they're asking, is there any SUS training support or knowledge management to facilitate technology transfer?
Okay. So that's the first part. Yeah.
Kristen, you can start.
Yeah, that's a tough one to answer. So there's tech transfer within a company. So from, for example, one site or one facility to another, and then there's sort of like a tech transfer from the supplier to the end user. So speaking to the second one, the supplier to the end user, we do have a single-use training support. What we found is that the suppliers are extremely supportive with coming in and doing in-class training with their components and assemblies and giving us tips of the trade. In addition to that, I always like to have somebody in-house who's very familiar with the single-use assemblies to be a trainer in that in-class training. So it's a combined supplier and user training. And of course, going with that is the knowledge management that Sarah has asked here as well. So it's not only transferring just how to use it, but why is it used? What information is available from the suppliers, and that has been done in-house to ensure that this is something we want to bring in. What is it used for? With respect to in-house tech transfers, we typically do a training such that, again, it starts off with knowledge management. What is this used for? Why? What is the product that you're using it with? And then we would go into the observed, assisted, performed type training that is all documented in our GMP lesson plans. And again, that's got to be in class.
Great. Is there anything you'd like to add, Dr. Fora?
No, I mean, I think I'm really happy, I mean, with Kristen's answers. So we developed, specific training package, and I would say there is no doubt that E-based learning tool is as well part of the solution as well to help customers for further support.
Excellent. So that seems like all the time we have. I would like to take this opportunity to thank our presenters, Kirsten Stralendorf and Dr. Helene Pora, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com. Thank you very much, everyone.
Thank you.
Thank you.